Hi, AP Bio. Today we're going to be talking about the relevance of graphing to AP Biology, but not just to AP Biology. We're going to talk about why it's important for your general life, which you might not think that it is, but I would argue that it's actually really important. And to start us off, I want you to look at the example that I have on the screen. So this is showing the profits of a company and the distribution of those profits based on quarterly uh, statistics or quarterly measurements and then yearly measurements. And so I want you to look at these two graphs and I want you to think about if you were a CEO who is trying to convince the shareholders of your company that the company had continued to increase in its profits every year since 2019, which of these graphs do you think you would use? Now, I have a feeling that you probably said the right-hand side, the right-hand graph. And that makes sense to me, and I would probably agree with you because the right-hand graph ends up looking like the profits of the company have continued to increase steadily, reliably, while the graph on the left doesn't show as reliable of an increase. So, from a perspective of a CEO, the graph on the right probably is more accurate and it shows the variability in the data, but the graph on the right shows the averages. And those averages show a different story. And this really is what graphing is about. Graphing is about stories. And your job is to figure out what that story is. When you look at a graph, I want you to pretend like you are an investigator. I want you to pretend like you are a detective. And your job is to try to figure out the missing pieces of the story that you've been given based on the information in the graph. Now, if you are following along, you should have a practice slideshow that's associated with this lesson, as well as a student document. That student document should be linked on Schoology, as well as in this slideshow. So how are you going to do this? How are you going to be a detective when it comes to learning and figuring out what a story is that a graph is trying to tell you? There are four main things that you need to pay attention to. First thing is, what are the variables that are being measured? This is the number one thing that you would need in order to be successful when it comes to graphing and experimental design analysis, is understanding what variables are being measured. If you can understand the variables being measured, you have a pretty good chance of understanding what the experiment is about and what the story is that's being told. You also need to understand the trends in the data. Now, trends in the data mean increasing, decreasing, or staying the same. And how have those increases and decreases changed over the course of the experiment or over the course of the observations? You then need to take this information, the variables and the trends in the data, and use it to try to work your way backwards and figure out what was the hypothesis that these scientists were using during their experiment. And this is a really common question in AP Bio, is to work backwards from hypotheses, or excuse me, from graphing to figure out what was the experimental design process for these researchers. And then step four is a couple of extra little pieces of information that we're going to talk about in a second. One thing I want to remind you of is these terms, independent variable, independent variable. These are really important terms to be familiar with. We've talked about them in AP Biology before, and at this point, I'm hoping you are familiar with these terms. The independent variable is the one that is controlled by the scientist. The dependent variable is the one that the scientist is observing and measuring. And typically, these go on the X and Y axis respectively. So the scientist in this case, for the graph of population change over time, the scientist chose the time that they were going to measure this experiment for. Sometimes you will technically have multiple independent variables, but there's going to be one independent variable that directly influences the dependent variable. And that's going to be the primary independent variable for the experiment. Remember, the dependent variable depends on the independent variable. It depends on what is changed within the independent variable. So it's usually easier to try to figure out what the dependent variable is first, and then figure out what the independent variable is. First type of graph that you're going to pay attention to. It's a pie chart. It's very straightforward. deceptively straightforward. Students often overthink pie charts. But when you're looking at a pie chart, you're looking at percentages of a whole. You're looking at the breakdown of an entire piece of information or an entire whole set. So this whole set that we're looking at here is water use, and it's broken down by the percentage of what is using the water. Now, again, whenever we're going through these graphs, these are the four things you should be paying attention to. I want you, for this pie chart, to consider what are the variables being measured, what are the trends in the data, what was the hypothesis that the scientist or the researcher or the observer could have had before they started the experiment or the observation, and then is there any extra information that you can, provide to help you understand this graph? That's the information that you should be putting into your document if you're working on the document that was assigned with this lesson. Now, I'm going to go over some examples of graphing in AP Biology that are maybe a little bit more challenging. And they're good examples of needing to do a lot of reading in order to understand what's going on in an AP Biology experiment. Okay. So for this example, we have a species of crow. And this species of crow is known to fly to different heights to drop seeds that will break open and expose a highly nutritious fruit. The seed, however, will not break open on the first try, so the bird will need to take many flights in order to break the seed open. And this is energetically costly for the bird. So take a look at this graph. This graph, I'm going to minimize my video for a second. This graph shows the average number of drops that the birds are going to need versus the total flight height. Also, looking at the average number of drops versus the total flight height compared to the drop height. So there's a lot going on here. You have two different colors. You have two different axes on the y-axis. So there's a lot happening here. But this is just a bar graph. And this is a really good example of a bar graph because bar graphs are primarily going to be used for comparing parts of a whole or for comparing a value between groups. Okay. So when I said comparing parts of a whole, usually that's going to be a pie chart. However, sometimes you can do a bar graph because the bar graph is going to show you distinct groupings and how those groupings compare to each other. So in this case, we're looking at drop height and how the different drop heights influence the average number of drops that a bird had to take and the total flight height that the bird made over time. Now, when I said extra information, if you're remembering this fourth piece here, extra information, one of the pieces of extra information that I was going to communicate and wanted you guys to be familiar with is the idea of hatch marks. Hatch marks are these little lines here between 2 and 3 and between 7 and 15. And these hatch marks represent a break in the measurements of a data set. They represent a change in the scaling of your data set. So for example, if you're looking at this graph, you go from an increment of 1, you go from 2 to 3. Then you go from 3 to 5 meters. And so what the hatch marks between 2 and 3 represent is that variance, that step up in the drop height is different from the rest of the scale. Same thing from going from 7 to 15. This is also showing that the scale is different here. And so this is something that you'll often see if there is not a lot of relevant data between these points, or sometimes it can be a little bit deceptive. And hatch marks you want to be careful about because sometimes people can get rid of data in that set. And so you need to go back and look at the experiment that they did and consider, okay, why was this data eliminated? Why do we not have a drop height for any of the data between 7 and 15 meters? Something that you want to consider. Now, looking at this graph, this is just a simple bar graph, but this is also a really good example of the kind of complexity that you might get in an AP Biology FRQ or MCQ. because you have two different y-axes. The left-hand y-axis is showing the average number of drops, and the right-hand y-axis is showing the total flight height. And so what you can see based on this information is that the orange bars represent that as you increase the drop height that the bird is dropping these seeds, the amount of times that the bird has to drop the seed decreases. So that's this orange bar right here. The average number of drops decreases as you increase the drop height. And the total flight height has this interesting curve. The total flight height, if you're dropping from a height of 2 meters over here, you're going to have to drop that seed so many times. You're going to have to drop that seed 55 times in order to break it open, which means that your total flight height is going to be really energetically costly. So there's a preferred height by the crows, and that's what this is showing here. Now, it's interesting because if you look at 15 meters, and this might be why the researchers chose to represent 15 meters here because it shows some interesting data. If you look at 15 meters, it shows that the total flight height is substantially larger if they are dropping from 15 meters. So if they're dropping the seed from higher up, they're not going to have to drop it as many times, but they're still going to be having to fly an increased amount of times or an increased amount of height. And so if you think about height in terms of the energy that a raven is going to have to expend on this exercise, then dropping from a height of 15 meters, even though you would think, oh, I'll just drop the seed from a higher height and then I'll have to break them open less, or I'll have to take fewer flights in order to break open the seed. That's not the case because there's this Goldilocks zone where the bird tends to drop the seed from. So this is just a really good example of the kind of complexity that you'll see on an AP Biology graph. Now, something that you might see, I don't see it very often, but something that you might see in AP Biology is a histogram. And a histogram looks an awful lot like a bar graph. If you see this histogram over here, it's going to look very similar to a bar graph, but there's some slight differences. If you notice, the bar graph here is showing comparison of groups. You're showing different classes and the number of girls in those classes. The histogram is showing a distribution of the data. It's showing that of the marks that were obtained, there was a possible range of 0 to 30 points, right? 0 to 30 points or 0 to 30 marks if you're English, right? And this is the number of students. And so it's showing that there's a distribution of the data. And the way that you can tell if something is a histogram versus a bar graph is that the histogram, the bars are right next to each other. For a bar graph, there's going to be space between them. So you need to be really careful. If you are drawing a bar graph, do not draw the bars right next to each other, because then the AP graders are going to look at that graph and they're going to think that you drew a histogram. And if your data is not best represented by showing a distribution like a histogram, then your data might not necessarily, you might not get the point on that point or on that FRQ because it's not the most accurate way to represent the data. So this is not something I see very often, but it's just something to be aware of. Histograms and bar graphs are showing very similar information, but not exactly the same. And in fact, often they can show a very wide difference in the information, right? In this case, we're showing distinct comparison between groups. And in this case, we're showing a range in the marks obtained. Now, again, as we're going through these graphs, you should be filling out that chart. You should be thinking about what are the hypotheses that the researcher could have come up with for this experiment? What are the variables that the researcher is using? And what is the methods of their collection? And so that's going to be what you're filling in that chart that was originally at the beginning of this lesson. So for a line graph, you're going to look at the typically for a line graph, you're tracking changes over periods of time and then growth in very specific increments. Now, the way to figure out if something should be drawn as a line graph is if you have a really big number of data points that you're going to include on your x-axis. So for example, you have here the age of these gophers. I think they're actually prairie dogs. These prairie dogs have ages all the way up to 26. So realistically, drawing 26 bars on a bar graph is not going to be very reasonable. It's not going to be a helpful graph. And so that's a really good indication that you should draw a line graph, is when you have a lot of data points that are going to go on your x-axis. Now, another way to think about drawing a line graph, or the reasons why you might draw a line graph, is for showing that continuous data. And that's what you have here. You have a spread of data over 1 to 26 months, and you're comparing the distance that males versus females moved from their original birthplace. So if you were thinking about the story that this graph tells, right? You're a detective, you're figuring out what is the story. You might look at this data and you might say, okay, well, this tells me that female gophers or female prairie dogs don't move as far from their birthplace after they're born. Males tend to move farther from their birthplace. Now, this, if you're looking at that graph or that chart that I'm asking you to fill out, you might notice that this particular experiment, it's hard to figure out what the control is. One of the things that you'll be asked in experimental design is to figure out what is the control group for an experiment. And up until this point in AP Biology, we've mostly had experiments that included a control. However, for this particular graph, there is no control group. And that's going to be true also for the previous bar graph of the ravens dropping the seeds. Because a controlled experiment is one where you, as the scientist, create a control group. And that control group is your comparison for your experiment. A controlled experiment is where you, as the scientist, are trying to control as many possible variables so that the only variable that's changing is the independent variable that you are changing as the scientist. Everything else is held constant so that they don't influence the dependent variable. Now, typically in ecological studies like this one and like the raven example, ecological studies will not have a control. Think about why that might be. Why would ecological studies not have a control? What do you think? Oops. If you said anything along the lines of, well, if you're looking and you're observing at a system like a biological system, a biome, an ecosystem, a community, you can't really control for all of the variables in a wild ecosystem. And so you often won't see a control group in ecological studies. So let's take a look at an example that is an ecological study, but it's slightly different than the ones that we've looked at so far. So this is a lot of background information. I'm just going to summarize what's going on here. Adult male guppies exhibit genetically determined spots, while juvenile and adult female guppies lack spots. Male and female guppies in this experiment were from genetically diverse populations, and they were collected from different streams and placed together in an isolated environment containing no predators. At this point, I want you to be thinking, is this a controlled experiment? Because we're not observing the organisms in their wild habitat, we are taking them into a controlled environment. The study population was maintained for several generations in an isolated area before being separated into two groups. One group was moved to an artificial pond containing a fish predator, while a second group was moved to an artificial pond containing no predators. The two groups went through several generations in their new environments, and at different times during the experiment, the mean number of spots per male adult guppy was determined as shown in the figure. And this figure is going to have error bars. Now, hopefully you remember error bars from our previous lessons, but if you are unfamiliar or if you're reviewing, error bars are going to be used for displaying the variability of a data set around an average. Okay, the variability of a data set around an average. So in this graph, what we have is we have the time between when the group was originally caught and put into the tank, when they were separated into groups, and when they introduced the predator. The dotted line represents the predator being present in the group. The solid line represents the predator being absent. So if you look at this data set, the average or the mean number of spots is going to have an error bar associated with it. Now, typically, when we've thought about error bars in the past, we've thought about them in the context of reliability. How reliable is your data based on the standard error of the mean or based on the standard deviation? However, for this graph, there's slightly something different happening here. Now, this is a little tricky, so I'm going to give you some time to do it. I want you to take some time to work on this and pause the video to answer this question. I want you to describe the change in genetic variation in the population between 0 and 6 months and provide reasoning for your description based on the means and the standard error of the means. Describe the change in genetic variation in these populations between 0 and 6 months. Did you pause the video? What I want you to notice here is that this is not necessarily about reliability. The data at time point 0 does not mean that their data is unreliable. And this is a really, really important thing to notice because we have primarily talked about error bars in the context of reliability. But in this case, what this is showing is the variance in this data point. There was an average of 10 spots on these guppies when they were originally captured. But there was a variation of between 12, almost 12, and 8 spots. And so there was a large amount of variation in the number of spots between the guppies. What we can see happened from 0 to 6 months is that the variation decreased. There was less variation. The guppies, on average, had closer to 12 spots, and there was less variation in that data set. This is not about accuracy or reliability in the data here. This is specifically about variability in the data that was collected. So that's an important thing to notice about error bars that you might not have noticed in the past when we've gone over examples of error bars. Now, we've gone over a lot at this point. I recommend taking a break and using this time to fill out any of the information on that chart. I've been going through this fairly quickly, so I really encourage you to take some time and make sure that you're filling out each of the spots on that chart on your assignment, because this is a lot of information to try to process at once. Now, additional type of graph that you're going to need for AP Biology is a scatter plot. And scatter plots are going to often look similar to line graphs. It's just that the dots aren't connected. But scatter plots are showing something different. They're usually used for comparing large numbers of data points that are not talking about time. Usually you're showing data points and the relationship between two variables that is not on a continuous spectrum. So for example, in this example, we have the number of seeds planted per meter squared and the average number of seeds that are produced per reproducing individual. So what you can see is that as the number of seeds planted per meter squared increases, you have these dots up here. Overall, there's a decrease in the number of seeds that are produced by this species. So that means that if there are a lot of this species in an area, they're not going to produce as many seeds. They're not going to waste the energy on producing seeds because they know that those seeds might not be able to be germinated. So this is just another example of a scatter plot. Now, sometimes you'll see a scatter plot, and this is kind of confusing, so pay attention. Sometimes you'll see a scatter plot that has a line going through it. That's still not a line graph, it's still a scatter plot. But now what you are showing is the trend line. Now, in AP Biology, you're not going to be asked to calculate any trend lines or any averages or any means in this particular way. However, it is something to be familiar with. And so I encourage you, if you are unfamiliar, to review trend lines in scatter plots. Now, in this particular example, we're looking at clutch sizes, and clutch is just the number of total eggs that a bird lays per each nesting attempt. And clutch size can vary depending on a lot of different variables in a bird's environment. So interestingly, this data kind of shows something similar to the last graph. Except this time, instead of talking about plants, we're talking about birds. So in this graph, you notice that as there are more females in a particular area, their clutch size decreases. So this is a really interesting connection that both plants and animals are doing something similar. Again, as a detective, thinking about what the story is that these people are trying to tell with their research. Now, everything that we've gone over so far has very likely been review for you from other classes. What we're going to talk about now is something that you may not have seen in other classes. So let's take a look at an example, okay? Let's say that you are gathering information about the distances between exoplanets and Earth and then their radius. And then you decide to use light years for distance and kilometers for the radius. This is the graph that you've created, okay? Distance versus the radius of exoplanets. So what you're looking at here is the distance of these exoplanets from Earth and then the radius of those exoplanets in kilometers. So you'll notice that as you graph the data, most of the exoplanets are going to have data around 5 to 30 meters, or excuse me, 5 to 30 million light years, right? They're going to be in that relative data set. So you first decide to plot the planets with distances smaller than 100 light years. But then you add planets that are millions of light years away from Earth. And so you have more distance between these exoplanets and Earth. So these orange dots are the new dots with the planets that are farther away from Earth. Now we've kept the same scale. We still have 0 to our maximum, and we've kept the same increments. But now we're increasing. Our scale just got so much bigger. We just have so much more space that we are trying to fit onto our graph. So if you look at this graph, what do you notice about this that makes the data difficult to interpret? Ignore this bottom part right here for right now. Don't worry about this for right now. Focus on this top part, this top graph. What do you notice about this data, the plot, that makes the data difficult to interpret? I want you to pay attention to what happened to the blue dots. Did you see it? Using this graph, the blue dots now all look like they are 0 million light years away from Earth. But that's not true because in the last graph, we know that these planets, the blue dots, are all variable in their distance away from Earth. So what is it that happened here? Well, because so many of our data points were very, very small, And then so many of our data points were really, really large. We ended up with a scale that was much too big. So how do we fix it? We fix it by using a logarithmic scale. So a logarithmic scale is when you create an x-axis that can show more data by multiplying the starting point by a fixed factor. Now, that's a lot of information. What does that mean? Well, it basically means that instead of increasing your x-axis by 1, 2, 3, 4, 5, or 2, 4, 6, 8, 10, or 5, 10, 15, 20, 30, 25, 30, instead of increasing by a fixed factor, you're going to increase by, or excuse me, instead of increasing by the same number each time, you're going to increase by a logarithmic value. So for example, here, your logarithmic scale is increasing by 1, but then your logarithmic scale is increasing by 10, 20, 30, 40, 50, all the way up to 100. And so your scale is not going to be exactly even, because when you're graphing based on a logarithmic scale, the distance between 100 and 200 right here is going to be different. Now, why does this matter? How does this apply to the exoplanet example? Well, check out what happens if I take this data and I now plot it on a logarithmic scale. Check out what happened. Now I can show both. I can show the small planets and their distance in light years from Earth. And I can show the large planets and their distance in light years from Earth. And I can show them both on the same graph by showing a logarithmic scale. So this is a tricky thing, and we're going to practice this when we come back and we come back together and we work on this together. But I wanted you to see what a logarithmic scale can do for your data. It's going to be able to let you plot data that has really big values, really big distances between your values. And that's going to be really valuable for when you're graphing big data sets. Now, the last type of graph that we're going to look at is called a box and whisker plot. And box and whisker plots are typically going to be used for showing the shape of a distribution, its central value, and its variability. Now, people often get confused about box and whisker plots versus error bars. Box and whisker plots and error bars are showing completely different sets of information. It's really important not to overlap the two because they're showing completely different sets of information. Now, there are 5 main components to a box and whisker plot. You have the upper extreme, the lower extreme, the upper quartile, the lower quartile, and then the median. The distance between your upper extreme and the maximum of your upper quartile is going to be your whisker. And the same thing for the lower quartile and the lower extreme. The box is going to be everything from the median to the upper quartile. Now, what does this even mean? What does this mean? Why does this matter? Well, in a box and whisker plot, you can show the variation and the variance of your data using percentages of a whole. And so you can show a whole value and show how that value or how that data set is broken up in a really interesting way. Now, the best way to think about a box and whisker plot is by thinking about a bell curve. And if you've ever taken statistics or you've looked at a bell curve, what a bell curve shows is the variation in a data set and how much of that variation falls within the norm or falls within the average or the median, right? So if we're looking at this data set here, our box of a box and whisker plot is going to be 50% of the data. Each whisker is going to be 25% of the data. All of this is really meaningless and really almost impossible to understand if you don't have an example. So let's look at an example first, because again, it's really hard to conceptualize a box and whisker plot without looking at an example first. So let's say we want to analyze the distribution of species richness of a field that has been cleared near the edge of a forest. And so I have a picture right here of what we're talking about. This is a forest. So on this side would be the forest. On this side is the field that has been cleared of trees, and there's a road separating. And so what we have over on the right is our box and whisker plot. So what I want you to do is look at this box and whisker plot and figure out, okay, where's my median? What's my upper quartile and my lower quartile? And what's my upper extreme and my lower extreme? Your median here is going to be 8. So what does that mean? What that is talking about is that of the fields you surveyed, let's say that you surveyed 100 fields. 8 of those fields had a species richness, or excuse me, not 8 of those fields. The median number of species richness meaning the number of species in that area, is 8 species. Let me say that again to clarify. When you've surveyed these fields, the average or the median number of species was 8 species. Now, 50% of the fields that you surveyed fell within 10 to 7 species. That's what the box represents. It represents 50% of your data. 50% of your data, meaning 50% of the fields surveyed, right? These fields had between 7 and 10 species. 25% of the fields surveyed had between 10 and 11 species, and 25% of the fields had between 5 and 7 species. That is what the data is representing. It's not simply analyzing one value, it's analyzing how 5 different values fit together and what the relationship is between them. So let's take a look at an example. Let's kind of question ourselves a little bit on this. Approximately what percentage of field surveyed falls above 7 species in terms of species richness? What percentage of the fields surveyed falls above 7 species in terms of species richness? Did you get it? 75% of the fields surveyed are above 7 species, because remember, each part of the box, each half or each section of the box represents 25%. That's what this is explaining here. 25% of the data falls in that lower quartile. 25% of the data falls in this upper quartile. And then 25% falls within that upper quartile to the upper extreme. So if you add those together, 25 plus 25 plus 25, you get 75% of the field surveyed had a species richness above 7 species. Now, this data is not as relevant if you aren't comparing it to something. So we're going to compare fields to near the field, to the very, very edge of the forest, to very near the forest, the edge of the forest, to within deeper in the forest. So these are 5 different locations. Whoops. 5 different locations. There's species richness, meaning the number of species that exist in that location, and the distribution of those data sets. So looking at this data, try to answer this question. What does it mean if the lower quartile is larger than the upper quartile? For example, in the forest, what does it mean if the lower quartile is larger than the upper quartile? If you said that this means there is more of a range in the data in the lower quartile than the upper quartile, meaning that 25% of your data falls between 9 species and 12 species, whereas 25% of the data here falls within 12 to 13 species, meaning that there are more locations of the forest that have a species richness between 12 and 13, then you would be correct. The lower quartile being larger than the upper quartile just means that there is a wider variation of data within this 25% of the data set. So that being said, we're going to come back together and we're going to practice using a lot of this information together. So go through that chart, try to fill out as much information as you can, and I'll see you in the next one.